Thank you for coming back. In this video, I would like to show you how can you annotate your variant files using ANNOVAR software, which is a online web server and user friendly. You can easily use it. So last time we annotate our VCF files, your mutation files using CGI, which is a cancer genome interpreter. So CGI gave you whether your mutation is a driver mutation or a passenger mutation based on cancer data. But if you want extra information, a lot of extra information that will help you in your research to prioritize your variant, to work more on your variants, to filter out the pathogenic variants, you need extra information. So basically the ANNOVAR software will help you with that. So yeah, using the same VCF file that we have used for CGI analysis, but remember CGI analysis only gave us which variant is driver, which is passenger, which is not enough information. We need extra information to prioritize our variant and to work more on our variants which are actually pathogenic and filter out the unimportant mutations, because usually in the research we are more focused on the damaging mutation that actually are damaging the protein-protein interactions that are and usually sometime we filter out mutation that are actually involved in the pathways that we are interested in. So yeah, so the extra information will really help us in that way. So again, this is the same VCF file, and when we see the VCF file, we don't have any information based on this VCF file. We only know on which chromosome the variant is present, what is the position where the variant is present, and then what is the reference allele and what is the alternative allele, what is the quality of sequencing, and based on filters that have been applied during sequencing, and all the filters that have been applied during the sequencing, whether that particular variant passed all the filters or not. So we need to annotate this VCF file, but as you can see, you can't clearly see this VCF file and information present in this VCF file. So what I usually do, I paste this VCF file in Excel to better look at my variant. So let's do that. So I pasted all the variants. Now I can clearly see everything. So this is your the chromosome number where your variant is present, and this is the position of your variant on that particular chromosome. And what is ID? So ID is basically when you give a specific ID to your variant. So I haven't given any specific ID to the variant, so that's why this column is empty. And then reference allele, so what is actually the reference allele there, and what is the mutation, which is alternative allele there. And quality is basically the sequencing quality for that variant. And then the filter is denoted by pass. Pass means the variant has passed all the filters, so it is good to go for analysis. So yeah, so we selected the variants which are denoted with pass. And then also info is giving you extra information about the sequencing, and then format. So format is basically again giving you information about your sequencing information like forward read, reverse read, allele frequency, and sequencing depth of your variant. So allele frequency, so what is allele frequency and what is the DP, which is important in the research. For example, for my PhD project, I worked on the allele frequency and DP. I filtered my variants based on allele frequency and depth. So the allele frequency threshold based on other researchers and other references, so I put 5%. 5%. So any variant which has allele frequency more than 5%, so I considered is like a valid variants. Other than they could be sequencing errors, so I just removed them. I filtered all the variants out. For DP, so my threshold based on other studies, I took 100. So if the variant depth or the variant is the read depth, so if the variant is present on 100 reads, that could be considered valid variant, not a sequencing error. So this is what I considered for my project. So here, so how to find what is the depth and allele frequency of the variant? So it is literally in the next column. So allele frequency is the third information. So the third information is basically the 0.993. so the allele frequency for this variant is 99%, and the depth is 234, which is literally next to allele frequency. So yeah, this is all information given in the VCF files, but let's annotate this file to get more information about your variants. So basically today we will be using the mutation files to annotate them, because we don't know anything about your RB mutation file. So if you want to learn this software, which is by WG Lab, so this is the lab that developed this software. So very good software this is. I really like this software. So if you want to learn how to use this software, you can go to tutorial. So they have given some useful information about this web server, this software. For example, what type of annotation you can get using these softwares, what input file you can use. For example, we will be using the VCF file, which is variant calling files. You can use the TSV files as well. And then you can get annotation based on phenotype and disease specific prioritization, but today we are not using that. And then another information, the parameter setting includes reference genome. So obviously when you are annotating your mutation files, you should specify to which reference genome, based on which reference genome you want annotation for your mutation. So we will be using HG19 like we did in CGI analysis, and again same input format is VCF file we will be using. So yeah, software. And when we go down, so there are some information they have given us, that keep page open after submitting a job. Yes, we will keep the page open, because some email servers treat the result email as spam email, and the results are deleted after 24 hours. So they are saying that keep the page open, so you can see the results, and the results will be there only for 24 hours. So you don't need to miss those information, and you don't want actually to miss those results. So here I will put my official email address, because usually this software needs the official email address, so they can just send the results to your official email. And sample identifier. So sample identifier means you can name your sample. So when they send you the results will be with that specific sample identifier. So you should remember that sample identifier. So usually I give the last 3 digits of my sample. So I will give L0065, I think it's L0065. So this is my sample, and now I will input the VCF file. Interesting thing is, like CGI, you can actually paste your variants in this section as well. You can just paste all your mutations, but the easiest way to do the ANNOVAR analysis is just input the file. So just go to the input file, and this is my VCF file, and open. After you upload your VCF file, if you go down to the parameter settings, so yes, you want the result duration 1 day, and then you have reference genome HG19. You have uploaded VCF file, and the gene definition is refseq gene, because that's this you need to select. individual analysis, yes, individual analysis, and then disease model, none, because I'm not selecting any disease model. So yeah, let's do the analysis, and then I will press, yeah, I agree to the terms of use. Yeah, it's already pressed there, so I will just submit. So after submitting, they will tell you that the date and the time you have submitted the job, and the results will be generated pressing this link. So I will press it and I will wait for the results. Okay, the results has been generated. So yeah, it took only I think 60 seconds, but the bigger the file is, the more time it will take. So the file was quite small, so that's why. So sample identifier, yeah, this is the name I have given to my sample, and then the file name, which VCF file I have uploaded, and file format, yes, it was VCF file, and the reference genome we have selected is HG19. And the disease model, no filtering, we haven't done any filtration, and the process variants, so the rows were 150. Okay, so now you have two type of output files. One is exome summary result and one is genome summary results. So exome summary results means it is giving you annotation for all the variants, those were in the exonic region, and the genome summary results is giving you information about all the variants, those are present in exonic as well as intronic regions. So you can download this file in the CSV file as well as in a text file as well, but I think the useful way is to just download in CSV file, because then you can open that CSV file in your R as well, easily you can open it, and you can just view it as well, because text file you can't easily view it, just like the VCF file, we had to paste that file into an Excel file to just look it properly. So let's view this file. So when you view this file, so you can download this file as well as you can view it online as well. So this is the variants present in the exonic region, which is a protein coding region. So let's see what information this file is giving me. So again, this file is also giving me the chromosome number on which the variant is present, the position, now the position is split into start and end, so it is very useful when it is about the frame shift mutations. So start and end the matters, and yeah, so when more than two bases are changed, yeah, it's useful in that case. And again, what is a reference allele and what is alternative allele, and on which region this variant is present, on which gene that variant is present, and what type of mutation it is, whether it is synonymous mutation, non-synonymous SNV, and whether it is a stop gain mutation, whether it is a frame shift mutation. So all the information is there. So this is interesting column. So it is basically summarizing you information about your mutation. For example, let's see the first one. So the mutation is in the gene FIK3CA. The transcript number, for example, the messenger RNA ID is this one, which is NM006218.4. and the exon on which the variant is present is 3, and the C mutation is where the base change, which is 363T, and the P means protein change. So isoleucine, which is I, at position 121 has been changed into again isoleucine, which is denoted by I. So this is just one mutation information, but there is if you see, some of the mutations are long lines with a lot of information, with a lot of NM numbers. So it is basically when your gene is transcribing into messenger RNA and alternative splicing happens, so one gene can give you more than one messenger RNA. So every messenger RNA got their own NM number. So the mutation will be in a different messenger RNA with different NM numbers. So it is same mutation but different NM numbers and different position, but the mutation will be different. It is only because of the different transcript of that genes. So yeah, so this is basically exon results. So let's bring this file again because this file contains extra information as well. Okay, so another thing is, honestly I'm not sure about this column, because I think they have recently updated the software and they have given extra information as well. So I'm not sure about this column. This is clinical data, so on which diseases this mutation was identified. So this column will give you that information. For example, this variant has been identified in inborn genetic diseases and PIK3CA related disorders. And in the next column there, this is interesting one. This is again a clinical information and the PubMed ID of the papers that has and those papers has talked about these variants. And then we have another column. Again, I'm not sure about this column. And the clinical significance column is very important, which I have used in my research in my PhD project, which is a clinical significance column. So means how significant this variant is in clinical study. So basically what researchers do, so they have identified a variant and they sometimes they work on that variant, and then they find whether this variant is associated with the disease or not, whether this variant is damaging the protein function or not, whether this variant is causing disease or not. So that is basically called clinical significance. So it is usually denoted by pathogenic, damaging, benign, or likely benign. So benign means that all the studies that have worked on this variant and they concluded that this variant is a benign, this variant is not causing disease, this variant is not damaging the protein function. Likely benign means some researchers are saying that this variant could cause disease, but some researchers are saying that this variant is not causing any disease. But there is another word which has been used is a pathogenic. Pathogenic means that based on all the researchers conclusion, this variant is basically damaging. Okay, so the other information ANNOVAR will give you is the pathogenic scoring. For example, in different studies, you might have seen the pathogenic scoring. Pathogenic scoring means how this variant is impacting protein function. So for that, different machine learning scoring has been used, but I haven't used these scoring in my study. So the popular ones are SIFT score, poly one, and another mutation tester you might have seen in some studies. So the scoring I have used is fat HMM scoring. So basically this scoring has outperformed the other scoring tools, so that's why I started using this scoring. But I haven't basically used this one because the problem with this one, I used the updated one, because the problem with this one is it can only give scoring to mutations give scoring for the mutation in exonic region, but I have both exonic and intronic variants, so I need a tool that can score both variants. So there is another updated version of that tool, which is this one, which is fat HMM MKL coding score. So this will give you information about both coding and non-coding variant scoring. So N means neutral, so according to this scoring, the variant is not impacting the function of the function of the protein, and D means damaging. That's according to this scoring prediction, your variant might be affecting the function of your protein. So I have used this in my study. So again, these are the scoring tools. Other interesting thing? So we have allele frequency from nomad. So nomad is basically a database that will give you the population frequency of for your variant. So what is population frequency? So basically when a variant is high frequency in a normal population, so basically we can't call that variant disease causing variant because it is like common in the normal population. So different studies use a set threshold to filter those common variants. So based on other studies that I have read, I used a score. I used the percentage 5%. So if my variant is present more than 5% in a normal population, I will filter that variant out because that is not disease causing variant, that is like a common variant. So that was my percentage in my study. So what other things we have? So okay, so the last columns are just the VCF file columns, so they have just given you the columns back. It is good as well because at least you can recognize these columns, and then if you have a specific identifier for that sample, you can easily recognize these columns, that this column belongs to this sample. So I think in that case ANNOVAR giving you back your VCF file columns is like very helpful. So that's all about this video. See the genome ones now. So when we view the genome summary result, so this is genome summary result. So if you see here, you are finding intronic mutations as well as exonic mutations. So genome is basically giving you information about all the mutations. But yeah, one thing I noted that in this file they have only given information about the intronic region, because I think according to them they have already given information in the other file, so they don't need to put the same information in this file as well, and the rest of the information is same. um, we can go back and then we can download simply clicking on this CSV file, and then your file will be downloaded here. So that's all about today's video. I hope I helped I helped you guys how to use this ANNOVAR software. And at last I would say that we are researchers, and every day is a school day. We are still learning at this stage, and so many things to learn, and so many things to improve. So if you have a question, or if I can help you with anything, just put me an email, and then I will try to help you. And in case of any other question, just put in the comment section. Thank you so much. Thank you. I hope I'll see you guys again. Bye.